Hello and welcome to today's webinar featuring Donna Werling. I'm Claire Cameron, Engagement Editor at Spectrum, the home for autism research, news and analysis, previously found on safari.org. We'll begin the presentation in a moment, but first, know that we'll be fielding questions at the end of today's presentation, but you can ask them at any point during the session. To ask a question, submit them via the chat window to the left of your screen. And as always, I'll note that if there are any members of the press tuning in, you can only report information presented during this webinar if that material has already been published elsewhere or if you have first obtained express written permission from the presenter. Donna Werling joins us today from San Francisco, California, where she is currently a postdoctoral scholar working jointly with Dr. Matthew State and Dr. Stephen Sanders in the Psychiatry Department at the University of California, San Francisco. Dr. Werling previously completed her PhD in neuroscience under Dr. Dan Geschwind at University of California, Los Angeles. Dr. Werling's work focuses on identifying and understanding the biological mechanisms that contribute to the development of autism, including the discovery and identification of risk genes for autism. In addition, Dr. Werling investigates how differences in the neurobiology of males and females may contribute to risk for autism. Dr. Werling has received several fellowships to support her training and research, including the University of California, Los Angeles Chancellor's Prize, a fellowship from the Achievement Rewards for College Scientists Foundation, a pre-doctoral Ruth L. Kirstein National Research Service Award, and most recently, a postdoctoral fellowship from the Autism Science Foundation. Welcome, Dr. Werling.
Thank you for that introduction, Claire, and thank you all for tuning in. I'm excited to be able to present to all of you today about the role of sex-differential biology and risk for autism spectrum disorder. I'm pretty confident that most of the people on the line are very familiar with what autism is, but I'll just run through a little intro slide here to get everyone up to speed. So autism spectrum disorders, or ASDs, are pervasive neurodevelopmental disorders that are characterized by deficits in social communication and interaction, and by restricted repetitive behavior, interests, or activities. The most recent prevalence estimates for ASD in the United States is 1 in 68 children, or approximately 1.5%. The cost of ASD, at least in the U.S., in terms of monetary costs and cost to quality of life for many individuals with autism and their caregivers, are enormous. And currently the best treatments that we have available for these individuals are behavioral interventions. Now these treatment approaches are wonderful, but they're incredibly time consuming, and it would be great if we could potentially augment these behavioral interventions or even replace them in some cases with pharmaceuticals, but we're just not quite there yet. One thing that we do know about autism biology, however, is that genetic factors contribute to autism risk. We also know that ASDs have a sex bias prevalence. So approximately 4 times as many males as females have an ASD diagnosis. And this male bias has been true of autism since the very beginning. 8 of the 11 cases that Leo Kanner described in his 1943 publication were males. And this male bias has remained consistent across time and across different countries. So the diagram that you're seeing here is a summary of male to female ratios on the x-axis here of a number of different epidemiological studies looking at autism prevalence in different countries at different times and using different variations on diagnostic criteria. This dotted line that you see here marks a male to female ratio of 1. And so you can see that although across these studies the magnitude of the male bias varies somewhat widely, the male to female ratio in every one of these studies is always to the right of this dotted line. There are always more males identified than females, demonstrating just how consistent this is. Now this male bias appears to also vary by intellectual ability. So if we look at autistic individuals who have normal to high IQ, the male to female ratio can be as high as 9:1. And if we look at individuals who are also intellectually disabled, the male to female ratio approaches something more equivalent and has been observed to be as low as 1.6:1. I'd like to point out that the studies that are summarized in this plot and the studies that have observed this very, very high male skew among normal to high IQ individuals largely come from examination of existing medical and education records. And I think it's worth mentioning that some more recent epidemiological studies, prevalence studies not shown in this diagram, that have used intensive screening methods across large general population samples have been able to identify a greater number of females in general, and particularly those with normal to high IQ. Now, even though the sex bias in these studies is less pronounced, it's still around or just over 2 to 1, so the male to female skew still remains in these cases. Now, we're finding over time that females' ASD symptoms may in fact differ from males in key ways. Like I just mentioned on the previous slide, females with autism diagnoses are more likely to also be intellectually disabled. And we're also finding that females may show fewer restricted interests than males, or restricted interests in different domains than males. And these observations bring up some critically important questions about ASD diagnoses. One of them being, are the diagnostic criteria for ASD biased toward a male phenotype? In other words, are the checklists and the instruments that we use to diagnose autism, do those better describe what autism looks like in a male than in a female? And are there autistic females out there who are not being diagnosed? Would they benefit from diagnoses and services that are available to individuals who have diagnoses? Now, these questions are absolutely critical ones to keep in mind. For me, for all of you listening, whenever you learn something about or hear something about sex and autism. And the answers to these questions are being studied, but they're not fully resolved. I'd just like to point out, though, that if the prevalence findings from intensive screening studies of autism are any indication, the harder we look for autistic females, the more we will find. But it does appear to be likely that the number of males with autism diagnoses will still outnumber females. That said, only time and additional data will tell us if that is the case. In the meantime, though, for this presentation, I would like to focus on a hypothesis regarding the role of biology in these differences. So it is true that there are biological differences between the male and female brain, and it is possible and likely that these sex differences contribute to the differences we see in ASD risk and presentation. We may find that they are not the whole story, but they're certainly likely to play an important role. Now, why is this something that we're interested in? It's clear that biological sex is a potent risk modulator for autism. I mean, purely from a statistical standpoint, if you are male, you're far more likely to have an autism diagnosis than if you're a female. And to the extent that this difference is based on biology, there's great potential here for developing treatments. So if we find, for example, that males are at increased risk for autism, maybe we can design a treatment to target and neutralize those male-specific risk factors. If we find that females are protected from autism, then maybe we can design a treatment that will mimic female protective mechanisms. Now, to get to the treatment development stage, we really need to understand how sex-differential biology acts to influence ASD risk. We need to understand the factors that are involved, yes, but we also need to thoroughly understand how they operate and how they work together. And unfortunately, we're not quite there yet, but I would like to take the rest of this presentation to give you all a little summary of some of the ways that this question of trying to sort out this mechanism is being addressed in the field. So I'm going to go over these 3 topics. The first, I'm going to introduce the female protective effect model. Then I'll discuss what is currently known, or some of what is known about the role of sex steroid hormones in autism risk. And then I'll finish up with a description of data from a study that I led in Dan Geschwin's lab at UCLA about sex-differential gene expression and what that can tell us about the pathways involved in autism risk. Now, each of these topics are interrelated but somewhat distinct, like 3 mini-talks. So if you find yourself dozing off during one section, maybe perk up for the next because it might be of more interest to you. I will start now with the female protective effect model. And to talk about this, I need to set the stage with some theoretical material, but I'll transition into more data as we go on. So to understand what the female protective effect model is, we need to first try to think about autism risk as a liability model. So a liability model says that liability, or the total risk that might contribute to risk for autism, is quantitative, and that it follows a distribution in the population, say a normal distribution. Under this model, then, there are a few individuals who have very, very low liability for autism and experience very little risk. And there are some individuals at the high end of the distribution who have very, very high liability for autism. There's been a threshold of liability beyond which individuals wind up with a diagnosis of autism. So if you have liability that scores more or less in this high range, you will have autism. That's the way this model works. But with a disorder like ASD, where males far outnumber females, we need to tweak this model to become what we call a multiple threshold liability model, where there are different thresholds of minimum liability that are sufficient to lead to an autism diagnosis in males than in females. The females' threshold is higher. Now, this model is also referred to as the female protective effect model. This is essentially a synonym for the multiple threshold liability model in this case, because it looks like there's this range of liability that is sufficient to cause autism in males, but not in females, suggesting that females are protected from risk factors of this severity, if you will. But is this true? Are females actually protected from risk, and how can we test for that? Now, we can test for that by using this model to make predictions about what we might observe in the real world and then test those predictions. And most of the predictions from the female protective effect model come from taking a close look at the highlighted part of the distribution here. So let's zoom in. Now remember that the x-axis here is x-axis of risk here is a quantitative scale. So under this model, if we're looking at females who are diagnosed with autism, we expect that their average liability is going to be higher than the average liability for males who have autism. So how do we test if this is true? One way to do that is to look at specific types of risk for autism. So instead of talking about liability or risk in the abstract, we can look at factors that we know influence autism risk. And the risk factor that we know the most about or understand the best for autism is genetic risk. So in this case now, we say if we're looking at a sample of individuals who have autism diagnoses, we expect the females will have higher genetic risk than the males will. To the extent that this genetic risk is shared with family members of autistic individuals, then we would also expect the relatives of diagnosed females to be at higher risk for autism themselves than the relatives of autistic males. And this pattern is called the Carter effect. Now there are a number of ways that we can, well, there's one typical way that scientists look for the Carter effect, and that is typically by looking at recurrence rates for autism in the younger siblings of individuals who already have an autism diagnosis. So what happens to this younger sibling? And we would expect to find that recurrence rates in the younger siblings of autistic sisters are higher than recurrence rates for the younger siblings of autistic brothers. So a couple of recent studies that have, or semi-recent studies that have tried to look at this include using a baby siblings paradigm where newborn younger siblings of autistic individuals are followed prospectively from birth to see if they wind up having autism later on. And this can also be done by looking at national registry data or any sort of registry data that tracks families and their medical diagnoses across time. So there are a couple of Scandinavian countries that are particularly great at this. One that I'm showing here is a study done on a Danish registry. There's also a similar study done in a Swedish registry. Now, the results from all of these studies generally follows the same tune, which is that highlighted here. They tend to find no main effect of proband gender in predicting ASD outcome. By that, they mean there's whether the younger sibling's older sibling with autism was a boy or a girl made no statistically significant difference on their chances of being autistic down the line, which is not really what we would hope to see if we're trying to prove that the female protective effect model is true. Here's another line of text from the, I believe this is the Danish study, again finding no statistically significant differences in risk dependent on the older sibling's sex. So all in all, these don't look too great for the female protective effect model. We'd expect to see that the sex of the older sibling does matter. However, a study from a couple of years ago used a different approach and found results that are consistent with the model. So here, instead of looking at categorical diagnoses, whether an individual has autism or not, these researchers looked at a quantitative measure of autistic traits. And they found that the siblings of females who scored very high on this measure, so we consider those the probands, also scored very high on this measure of autism traits, and higher than the siblings of males who scored highly on this trait. So this is consistent with the female protective effect model, and they found the same pattern in two independent cohorts of siblings, one from the UK and one from Sweden. Patterns from multiplex families from the AGREE cohort of autism families do also appear to support the female protective effect model. So multiplex families are families where there are two or more kids with autism. And this was a study that I did, again, in Dan Geschman's lab at UCLA, where I looked at the recurrence risk to younger siblings in these multiplex families. And in this case, I did find that there was a significant effect of whether at least one of these older siblings was female on the younger siblings' recurrence risk. So if there was a significantly higher recurrence rate for younger siblings of autistic sisters than for siblings who only had older autistic brothers. Now, additionally, instead of using siblings' recurrence rates to estimate how much genetic risk autistic males and females carry, we can also now look at the genetics of autistic patients to see if there's a sex difference there. Now, under the female protective effect model, right, we predict that ASD females have higher genetic liability than males do. But what does this look like? Well, this could mean that females have a greater number of harmful mutations in their genome than males do, or it might mean that females have larger or more harmful mutations in their genome than males do. And we can look for this now. So one type of genetic variant that has been looked for in this way is a copy number variant. So this is when entire sections of a chromosome are either deleted or duplicated. These genetic variants can be inherited from a parent, or they can occur de novo, or brand new, in a sperm or egg cell. And when researchers looked for these copy number variants in autistic kids from the Simon Simplex collection, they found that there was a higher incidence of de novo CNVs in autistic females than in autistic males. And they also found that these CNVs impacted a greater number of genes in the females than in the males. When researchers look at indel variants, or insertion-deletion variants, where just a handful of base pairs are inserted into the genome or deleted, I found a similar pattern, which is that females, shown in purple, have a higher incidence of this kind of harmful mutation than the males do. And when they looked at single nucleotide variants, such as single base pair changes that impact proteins, they again found a higher incidence of these de novo SNVs in high-confidence autism genes in the females compared with males. So together, these genetic data offer arguably more direct and more consistent observations that do support the female protective effect model. So that's food for thought. So to summarize this section here, we see that females with diagnosed autism show greater genetic liability than diagnosed males, in that females tend to have more frequent, larger, and more deleterious genetic variants. And this pattern suggests that females are protected from less deleterious variants or risk factors, particularly those in this range of the liability distribution. And the Carter effect, or our ability to see whether siblings of autistic girls have a higher risk for autism themselves, remains an ongoing question. And while I do wish this story wrapped up cleanly from all angles, it simply doesn't yet. However, I will argue that what we see in the genetic data, when we look directly at the genetics of autistic individuals, is potentially more important and more convincing that the female protective effect model is fairly accurate and useful for thinking about autism risk down the line. So the next question then is, if the female protective effect is perhaps true, then what is responsible for this protective effect? And one of the factors that has been investigated in that regard are sex steroid hormones. Now, sex steroid hormones belong to this class of proposed mechanisms for sex differential risk, in that they are a biological factor that is very clearly different between males and females. And several other related factors have been proposed over the years to play a role in autism, including the hypothesis that autism is an X-linked condition. In this case, females are protected by having a second copy of the X chromosome. It's also been proposed that there are genes on the X chromosome that are protective, but these genes are only expressed from the X that is inherited from the father. So since males inherit a Y chromosome from their father, not the X, they don't have these protective genes. And well, there are certainly genes on the X chromosome that contribute to autism risk. Genetic studies show that risk genes span the genome. They're autosomal, they're on the X. So it doesn't appear that X linkage can account for everything. And such protective genes as have been proposed on the X chromosome also have not yet been identified. They may still be out there, but we haven't found them yet. And then the third one, our third factor here listed in this table, has to do with sex steroid hormones, particularly fetal testosterone exposure, and that's the one that I'll focus on here. So this came to the forefront of the field's attention as part of the extreme male brain theory, which is a theory proposed by Simon Baron-Cohen back in 2002, that describes autism as an extreme or exaggerated version of male-typical cognitive and behavioral traits. Now, since testosterone is the biological factor that is largely responsible for directing the development of a male brain and body in utero, then it makes logical sense to propose that perhaps testosterone also acts to increase ASD risk under this model. Now, the Baron-Cohen group has followed up this hypothesis in a couple of different ways. One that I'm showing here, it comes from a study where they recruited mothers who had had amniocentesis during their pregnancy. They were able to get a sample of the amniotic fluid and measure the level of testosterone in that fluid to try to determine how much testosterone their children had been exposed to in the womb. They also sent these mothers a questionnaire called the Autism Quotient that they filled out about their child. And then what they observed was a significant positive correlation between fetal testosterone levels and scores on the Autism Quotient. So the higher your testosterone level, the higher, in general, your score on the Autism Quotient. So this is interesting. It does appear to support the potential existence of a relationship between autism traits and testosterone exposure. But that said, it's not, this distribution of points across the range of testosterone is pretty wide, and across the range of autism-like traits is pretty wide. So it's not as though we can take an amniocentesis sample, measure the testosterone, and predict that child's later risk of autism. But at the group level, there appears to be a relationship here. And since then, this group has moved on to look at the relationship between testosterone levels in utero and later autism diagnoses. So not just a quantitative measure of autism traits, but whether these individuals actually were diagnosed. And here now, testosterone levels are shown on the y-axis. This study, again, took advantage of the wonderful record-keeping and biobanking of the Danish. And you see in pink or red here, controls, and then 3 groups of individuals with autism-related conditions, PDD-NOS, Asperger's syndrome, and childhood autism. And what you see here is that the average, the mean, shown by these black dots, of testosterone levels in these autism groups in the cases is higher. It's elevated above that of controls, suggesting that there may be a relationship, again, between testosterone levels and autism diagnoses. But of course, as you can see, the distributions of testosterone levels in controls and cases overlap substantially. So again, though we cannot predict someone's autism risk based on their testosterone levels, when we put large numbers of samples together, these patterns appear to emerge. Now, that said, the researchers who did this study also looked at a number of additional steroid hormones beyond just testosterone, including progesterone, androstenedione, and cortisol, the hormone involved in our body's stress responses. And they find the same pattern in all of these other hormones, in that they are elevated in the autistic cases as compared with the controls. So while this study seems to support a relationship between testosterone and autism, it also calls into question the specificity of testosterone's role in this process. And this suggests several possibilities. I mean, it may still be the case that testosterone is the key factor, and elevation of these other hormones is simply a side effect or confound. Or it may be that one of these other hormones is the key, maybe cortisol. Or it may be the case that elevated steroidogenic activity or elevated steroid biosynthesis activity during fetal development in general is a risk factor. And further work is needed to sort that out. Now, the Baron-Cohen group has also applied a different approach for investigating hormonal activity and regulation in ASD, and this plot that I'm showing comes from a study focused on women with autism. So here they recruited over 400 high-functioning women with autism, so they were able to self-report on their own symptoms. And they asked these women about any co-occurring medical phenotypes or other traits that have been shown at some point in time to be associated with hormonal activity or testosterone exposure, including sex-linked conditions, reproductive conditions, timing of puberty, and gender and sexuality. And they found that a large number of these conditions or traits are more frequent in women with ASD than in controls, including anorexia, epilepsy, irregular menstrual cycles, excessive menstrual bleeding, precocious puberty, tomboyism, gender dysphoria, transgender identity, and homosexual, bisexual, and asexual sexual orientation. Now, it's not clear from these observations whether testosterone exposure during fetal development is the key for these differences, or whether hormonal regulation across the lifespan is related to autism symptoms. But these observations are consistent with the idea that maybe atypical hormonal regulation may have something to do with autism risk. So to summarize this section, results from studies show that fetal testosterone levels are associated with autism traits and diagnoses, but testosterone is not the only steroid hormone that's associated with ASD, and this requires further investigation. We see that females with ASD show symptoms of atypical sex steroid hormone regulation, and again, this suggests that the relationship between sex steroid hormones and ASD may extend beyond fetal exposure levels. All right, but so even if we accept the possibility that hormones are involved in autism risk, by what mechanism do these hormones act to affect the brain and to affect autism risk? And one study that I'll share with you that I led trying to address this question involves sex-differential gene expression in the brain. Now, I would classify the study of sex steroid hormones and these other sexually dimorphic biological factors as candidate mechanism approaches. So for these approaches, we take a biological factor that is known to differ between males and females, and then follow up on this pathway or this process in development, and to see if these downstream consequences of these biological factors intersect with autism biology in some way. So the advantage of these approaches is that we know exactly where these pathways start. We have great confidence in the fact that they are sexually dimorphic, but we don't necessarily know where each of these processes will wind up and if their effects downstream will intersect with autism biology in meaningful ways. So an alternative instead is to attempt to look at all of the pathways in the brain or body at once, and then try to see where sex-related pathways and autism-related pathways intersect. And one of the ways that we can attempt to do this is by looking at gene expression genome-wide. So the approach that I used for this study goes like this. So we want to, first of all, identify genes that have sex-differential expression levels in the brain, genes that show higher levels in males or higher levels in females. And we also want to compile sets of genes that are associated with autism. These could be genes that have mutations in them in autistic cases, or they could be genes that show differences in expression levels in the autistic brain compared with controls, for example. And then we look to see where these gene sets intersect. Do they intersect? Does this intersection implicate specific aspects of biology, and are those interesting? So this simple overlap approach can be used to test a couple of different hypotheses. The first is what I'll call the direct hypothesis. So this hypothesis recognizes that sex-differential regulatory mechanisms, for example, the binding of estrogen receptors in the genome or androgen receptors, for example, impacts neurodevelopment, impacts molecular pathways, cellular function, neural circuits, and so on. But perhaps maybe some of the targets of this sex-differential regulation are autism risk genes themselves. And it's because these genes are used differently in the male and female brain that you wind up with a sex difference in the prevalence of ASD downstream. Now, another possibility is what I'll call the indirect hypothesis, where we again recognize that sex-differential regulatory mechanisms impact neurodevelopment in several different ways, but we also know that autism risk genes don't operate in a vacuum. They're influencing neurodevelopment as well in the same types of processes as sex-differential regulation. And perhaps there is some intersection between the targets of sex-differential regulation and the pathways that are impacted downstream in autism. And it's at this point of intersection that you get the modulation of autism risk. So we test these hypotheses by comparing gene lists that correspond to each of these categories. So for example, the direct hypothesis, we look at genes that are sex-differentially expressed, and we look at autism risk genes. Is the overlap between these genes what we expect? Is it less than what we expect? Is it greater than what we expect? Same for the indirect hypothesis. This time we look again at genes that appear to be sex-differentially expressed, and then genes that are indirectly associated with autism or downstream associated with autism. Again, we ask, is the overlap between these gene sets what we expect, less than what we expect, or greater than what we expect? And a greater than expected overlap between these gene sets would be positive evidence for the hypothesis, suggesting that it may in fact be true. But to start to ask these questions, we first need to identify genes with sex-differential expression in the brain. So to do this, we made use of the BRAINSPAN data set. This is a publicly available data set that has gene expression data from the post-mortem human brain across development, starting from about 4 weeks post-conception up through late adulthood. And there are multiple brain regions sampled per individual brain with gene expression data. And we use these data to look first at the adolescent and adult brain, and then later I'll show you data also from the prenatal brain. Now the main data figure that I will be showing you is a heat map, so I want to introduce you to what that means when you look at it. So the top row of every heat map is going to be showing you or representing genes that have higher expression in the male brain, and I'll refer to those as male DE genes or male differentially expressed genes. The bottom row of each heat map is showing genes that have higher expression in females, and I'll call these female DE genes. Every column of the heat map represents a different set of autism-related genes. Now in each cell of this table is information representing the overlap between the row gene set, so the male DE gene set in this case, and the column gene set, so ASD gene set 1, for example. Now if this overlap in some of these cells is greater than what we expect by chance, this will be marked by a red circle, akin to this scale over here for enrichment and depletion. And if the overlap is less than what we expect by chance, it will be marked by a blue circle, kind of like this. So hopefully that makes sense to everyone on the line. And we'll jump into the data now. So here's a heat map schematic. Again, male DE genes represented in the top row, female DE genes in the bottom row. And then we've tested here 4 sets of genes that have shown evidence that they are directly associated with autism risk. So we have candidate genes from early genetic studies, genes that have protein disrupting rare de novo variants in autism cases, genes with missense variants, and genes that are targets of the FMRP gene. FMRP is a gene that causes Fragile X syndrome, a syndrome that has substantial overlap with autism symptoms. And this gene regulates the translation of a number of different transcripts in the genome. And so these targets have been shown time and again to be strongly associated with autism risk, so we include it here. Now, I actually don't have any screen to lift or any data to reveal on the slide because not one of these comparisons deviated from expectation. So we don't see any significant enrichment of male DE genes with any of these directly associated genes or female DE genes. So this evidence does not appear to support our direct hypothesis. So next we started to look at gene sets that are indirectly associated with autism. And to do this, we turned to results from a study done by Irina Vonigo and Dan Geschwin's lab, where she looked at gene expression data in the autistic brain and compared it to controls. So here now we start to see some results. So this first set of genes are genes that showed higher expression in the autism brain compared with controls. And then here are genes that showed lower expression. What we see is that male DE genes overlap significantly more than we expect by chance with genes that are upregulated in the autism brain. And the number in the middle of the circle is the p-value of that enrichment. We also looked at gene co-expression modules from the same study of expression in the autism brain. And a co-expression module is a set of genes that show similar expression levels. They tend to go up together, go down together across samples. And these sets of genes you tend to be able to annotate the functions of these sets of genes to common functions. So genes that are working together to accomplish the same task tend to show similar expression levels, and that's what a module is. Now, the vast majority of modules identified by this study behave very similarly in the case and control brain, in the autism brain and control brain. But there were two modules that behave differently in the autistic brain. So one of these is what we call the M12 module. This module showed lower expression in autism compared with controls, and the genes in this set were enriched for neuronal and synaptic function. And then this second module, we call this the M16 module. This module showed higher expression in the autistic brain compared with controls, and this gene set was enriched for immune functions, inflammatory functions of the brain. Now we see that the male DE genes overlap significantly with this M16 module, and the female DE genes overlap significantly with this M12 module. We also looked at co-expression modules from a more recent study of gene expression in the autism brain by Gupta et al. Out of Don Arking's lab. And here we find that male DE genes show significantly less overlap than we expect with this one module, ASD-M1. Now, we looked at an independent sample of adult brains and looked at their sex-differential gene expression, and we observed similar patterns to what you see here, and I won't show you those data, but I will show you results from the prenatal brain, which we also looked at. Now here, like for the adult in this section, we again see no significant enrichment of sex-differentially expressed genes with these gene sets that are directly associated with autism. We do see significantly less overlap than we expect between male DE genes and FMRP interacting genes, but no enrichment. Now we do see patterns that agree with the adult data. For example, male DE genes overlap significantly with genes upregulated in autism with the M16 module. And now in the prenatal data, we also see enrichment for a module from the Gupta study, this ASD-M5 module. Interestingly, both the M16 module and the ASD-M5 module, these two modules that are enriched for male expression, are associated with the functions of microglia cells. So microglia are not neurons. They do not transmit information across the brain. Instead, they are the resident immune cells of the brain. And this is particularly interesting because many of the genes that are directly associated with ASD risk function in neurons, especially at the synapse. So these results point to a different cell type entirely, suggesting that microglia may also have a role in autism risk, and particularly the aspect of autism risk mechanisms that is sex differential. Now, to drive this point home, we also compared our sets of genes that showed differential expression in the brain with sets of genes that mark the functions of different neural cell types from several different studies. So there's 1, 2, 3 here. And we find that in both the adult and prenatal brain, male DE genes are enriched for microglia markers from this study and from this study. And so there's agreement here. The adult data also shows some signal for astrocyte genes. This is another type of glial cell in the brain. And the prenatal data shows enrichment for endothelial cells as well. So if I can summarize this section, we find that genes that are directly associated with ASD risk are not differentially expressed by sex, at least in the adult or prenatal cortex of the samples we examined, suggesting that our direct hypothesis may not necessarily be true. And we're finding more evidence to support this indirect hypothesis that processes downstream from key sex differential factors and from maybe autism risk genes themselves intersect with one another. And this intersection may have something to do with the functions of microglia and possibly astrocytes, at least in the adult brain. Now, these cell types have already been implicated in autism by gene expression analyses. So the M16 module and M5 module from those 2 studies of gene expression suggest that these cell types may be doing something different in the autistic brain than control. These cell types may also be sexually dimorphic, not only according to gene expression from our analysis, but also according to a body of work from neurobiologists and neuroendocrinologists, mainly looking at rodent models, so rats and mice. But they find that the brains of male rodents are colonized by a greater number of microglia than females are, and also that astrocytes in the hypothalamus, at least, of males are larger and tend to show more branched morphology than the astrocytes of females. So there is some evidence here that the sex difference in these cell types may in fact be true. And these results now suggest that these cells may have a role in the neural mechanisms that drive sex differential risk for ASD. So I'd like to summarize each of the 3 topics that I presented here and where we stand more or less in our understanding of sex differential biological risk for ASD. So we know that ASD prevalence and presentation differ by sex, and we hypothesize that sex differential biology is likely to contribute, though while we recognize that it may not be the whole story, it's certainly likely to contribute. Genetic evidence in particular demonstrates the existence of a female protective effect in autism. There's evidence about suggesting that the regulation of testosterone and possibly other sex steroid hormones as well are associated with autism risk and traits. And then gene expression patterns in the human brain suggest that microglia and possibly astrocytes may participate in sex differential risk for ASD. Now, I know I mentioned at the very beginning of this presentation that it's not simply going to be enough to identify factors involved in the sex differential biology and risk, factors such as testosterone, factors such as these cell types, and that we need to better understand the relationships between these factors and the mechanisms that translate one step in the pathway to the next. So there's a lot of open road here to be explored. There are many gaps that need to be filled in before we can fully understand the mechanisms that are involved. And on this last slide here, I would just like to, I've tried to come up with a list of potential next steps of things that we can study to try to fill in those gaps. There are many more things that can be done, but here are a few. So with regard to steroid hormones, we should continue to explore the role of testosterone and other steroid hormones in neurodevelopment generally and in autism specifically. For gene expression, we need to replicate the patterns that I've presented here in larger independent samples, just to be sure that what we're seeing is true. And we also need to work to identify the cause of this male-female expression difference in microglia and astrocyte genes. Is it simply that males have a greater number of microglia in their brain, or do their microglia, are they more active, for example? We don't know. We'll also need to understand the consequences of sexually dimorphic microglia and astrocytes. What does that mean for neural connectivity and neural circuitry? And lastly, it would also be great if we could build up a more comprehensive understanding of sexual dimorphism in the brain and across the genome. So, and this knowledge will be very useful for putting findings related to autism in context. So it would be great if we could characterize the regulatory targets of estrogen receptors, androgen receptors, and other hormones, as well as transcription factor genes from the X and Y chromosome. And it would also be very useful to characterize sexual dimorphisms in brain regions beyond the hypothalamus. So the hypothalamus is a brain region that has been studied in great detail by endocrinologists and biologists. Because this region is responsible for reproductive behavior, it's also highly sexually dimorphic. But there may be perhaps more subtle sexual dimorphisms in other regions of the brain beyond the hypothalamus, and these regions may be more directly relevant to neuropsychiatric conditions such as autism that show a sex bias in their prevalence. And if we had a better understanding of the fundamentals behind those brain regions, then we might be able to understand why risk differs by sex. And with that, I'd like to thank my mentors here at UCSF, my mentors and colleagues at UCLA, my sources of data for this last study that I presented to you, and funding. And of course, thank you to Spectrum for putting this on for all of us and allowing me to present to you today. And thank you for tuning in. I'm happy to take any questions.
Okay, thank you very much, Donna. Our first question comes from Lise Elliott, who asks, is there data to address that you know of whether differential environment is a factor in the female protective effect?
That's a great question. And when we start to talk about environment, it's important to define what we mean by environment. So environment can mean many things. It could mean the way, for example, diet or chemicals that a child is exposed to, or it could mean things like social environment. So there is evidence out there to suggest that male and female babies are treated differently by their parents from the second they come out of the womb, and to the extent that that social difference, socialization difference, affects females and makes them either protected or appear to be protected, it is not necessarily well known. So I'm not quite sure what aspect of environment you're asking about, but there are very interesting studies out there about what is called, what has been named camouflaging behavior. So particularly for women with autism who are of normal to high IQ, there is evidence from self-report and studies that these women are able to, when they're out in the world, mask or camouflage their social symptoms such that they are able to fit in for a certain amount of time, but this is a very stressful process for them. But to the extent that that is caused by an innate drive to try to be more social, to try to fit in, or by an environmental push for girls to be more sociable, is unclear. But if you look up camouflaging behavior in autism, maybe that will address some of this question.
Okay, we have another question from Lilia Ikokeva. I'm sorry if I just pronounced your name incorrectly. Could overlap of M16 with male DE be due to the fact that M16 is capturing sex differences already, i.e. more male brains were analyzed in the Vonnegut paper?
Yeah, so this is a great question that I think I get asked every time I present these data. And the fact is that both the Vonnegut study and the Gupta et al. Study controlled for sex in their statistical analyses. So I don't think that's the case. But also, perhaps even more importantly than that, both at least in the Vonnegut study, the number of males and females, or the proportion of males and females in both the case and control groups were nearly equivalent. So it wasn't the case that, for example, the autism group had a greater proportion of male brains than the female or than the control group did, which would lead, like is being suggested, a conflation between male status and autism status. But if you look at the samples that were involved, there's actually approximately 25% of both the case and controlled groups were female. So while that might be contributing, I don't really think that that's the case.
Okay. We have another question from Dory Flores, who asks, has greater genetic liability in females been shown in low and high functioning individuals, or is it more characteristic of low functioning females?
Okay, another great question. So the genetic data that I presented comes almost exclusively from study of the Simon Simplex collection. So that's a collection of families that have only one autistic child per family. And so already you're looking at a set of families that may not necessarily be perfectly representative of the general population. And the individuals in that set, there's not enough females to be able to stratify in the way that you're suggesting between high functioning or high IQ individuals and those with intellectual disability. I will say, though, that our ability to identify harmful, deleterious genetic mutations is somewhat biased toward individuals who are cognitively disabled. So there's some skew there. But the bottom line is that we simply don't have enough samples available yet to be able to look at females in particular and separate out the genetic data between high IQ and low IQ individuals. But that would be something that would be really interesting to look at.
Okay, great. I think that is all the time that we have for questions at the moment. So thank you, everybody, for tuning in. And if you didn't get a chance to ask your question, please know that you will have an opportunity to do so. A complete replay of the presentation will be available on the Spectrum News site in a couple of days. You'll be able to ask questions in the comment section of that replay, and we'll be able to field them to Dr. Werling after that. So this webinar is part of an ongoing series by Spectrum. Our next webinar is in July with Rebecca Sachs. So please check our site for the upcoming talks. More will be announced soon, and for video archives of previous webinars. Thanks again, Dr. Werling, and thank you all for tuning in.